The overall goal of this technique is to provide a collection of protocols that are simple to carry out and can help any investigator characterize a given clinical or environmental isolate of Vibrio cholerae.
This method can help answer key questions in the bacteriology field, such as the specific genotypic and phenotypic characteristics of any Vibrio cholerae isolate, especially those of clinical relevance. The main advantage of this technique is that it allows any investigator to use this collection of assays to reliably characterize and distinguish clinical or environmental Vibrio cholerae isolates.
Together, these assays provide a systematic approach to be used as an alternative or in addition to costly labor-intensive experiments in the characterization of Vibrio cholerae clinical and environmental isolates.
Though this method can provide insight into the characterization of Vibrio cholerae biotypes, it can also be applied to characterize the biochemical and metabolic capabilities of other enteric bacteria.
Generally, individuals new to this method will struggle with time management as the incubation times necessary to ensure optimal results differ between biochemical assays.
Visual demonstrations of the spotting and stabbing protocols are critical as splatter can often lead to cross-contamination, and maximal spacing is required to prevent merging between cultures.
Begin the protocol by pelleting 1.8 milliliters of liquid overnight culture for 2 minutes by centrifugation at a minimum of 8,600 times G in a sterile 2 milliliter microcentrifuge tube. After centrifugation, remove the supernatant and resuspend the cell pellet in 900 microliters of fresh Luria bertani or LB broth by pipetting. Next, add 900 microliters of sterile 60